You may get gas. You don't always get gas, but you can get gas as a byproduct of fermentation. So remember, these were the ones that had those little upside down Durham's tubes in there. So if it is a gas producer, a little bit of that gas will get trapped in that upside down Durham's tube, okay? So that's how we assess whether it's acid and gas or acid and no gas. The other thing that can happen is if it's using the peptone, the medium will become basic. That causes the phenol red to turn a deep pink color. So when you see that deep pink, you know that's evidence of peptone utilization, not fermentation. Okay, so those are the two options. Either using the peptone or fermenting whatever sugar is in that test tube. So when you come back, you look at the color, and then you look in that Durham's tube to see if there's a bubble in there. So let's say your glucose tube turned yellow with a bubble. So you would read that as acid and gas. Usually in our lab notebooks, we record that as a capital A over a capital G. So that's for acid and gas. You can get acid with no bubble. Usually we record that as A over Ng, or either just A over a little negative, meaning that there was no gas present. And then if it's deep pink, in this case, this would mean it's not using the lactose. It's using the peptone. And usually we represent that as a capital K for alkaline. So that's how you would read that. So if you wanted to test mannose, you would set up another one with mannose. If you wanted to test xylitol, you would set up another one with xylitol. So each one is independent of all the others. So you're looking for that yellow color to see if it's a fermenter, and then you look in that Durham's tube to see if it's a gas producer or not. Okay, so any questions about that one? Okay. The oxidase test, that's the one that you add the reagent to and you get the purple color. The key to that one is you have to get the purple color within 20 seconds. Okay. Everything will turn purple eventually. You got to see it within 20 seconds to count that as a positive. So if you get that purple color within 20 seconds, you know that oxidase was present. Oxidase is one of the enzymes that's part of the electron transport chain, the respiratory system. So this tells you that it's actually cytochrome C oxidase, but we usually just shorten it and call it oxidase. So oxidase is present if you get the purple. If you don't get that purple within 20 seconds, that's a negative. Okay, so that one's pretty straightforward. The catalase test, we talked about that one earlier. You remember catalase is the enzyme that breaks hydrogen peroxide down into water and oxygen. So all of our cells produce catalase. So that's why when you use hydrogen peroxide to put on like a wound or a scrape or something, you always see a bubble. So what you're actually seeing is this equation. So when you drop that hydrogen peroxide on that wound or that cut, catalase is present, it breaks it down into water and oxygen. So this is what you see bubbling off. This is what actually makes those bubbles. So with microorganisms, a lot of them are catalase producers. So you simply drop hydrogen peroxide on there, you get bubbles, that's a positive. That means catalase was present. If you drop the hydrogen peroxide on there, there's no bubbles, that would be a negative. Okay? So both of those are pretty straightforward. So any questions about the oxidase or the catalase? We set up the nitrate test. So for the nitrate, it was specifically for nitrate reductase. Now, this was a little more complicated because you got lots of possibilities when you're talking about nitrate reduction. So the enzyme primarily is nitrate reductase. Nitrate reductase reduces nitrate to nitrite. But that's not the only thing that can happen. Remember, in the nitrate test tube, okay, this also has a Durham's tube in there, right? So it's got the little upside down tube in there. So that's the first thing we look at is that Durham's tube to see if there's a bubble in there. Now the key to this one is you have to know that your organism is not a fermenter. If it's a fermenter, that bubble in the Durham's tube could be something other than nitrogen gas, okay? So you've got to know that your organism is not a fermenter in order to know that that bubble in the Durham's tube is nitrogen gas. So that's one of the things that can happen. Nitrate can be broken all the way down into nitrogen gas. And a little bit of that nitrogen gas gets trapped in that upside down Durham's tube. But again, the key is you gotta know it's not a fermenter. It should be not a fermenter, not a fermenter. Too many blankets. Not a fermenter. Okay. So that's one of the things that can happen. And if that was the case, you'd be done at that point because that's a positive. So you know the nitrate's been converted all the way down to nitrogen gas. Okay? If there is no bubble, you can't say that's negative because the nitrate could have been broken down into something other than nitrogen gas. So it could have stopped short of that. So this is where you add the reagents. Remember we had the nitrate A and the nitrate B? We added them right after each other. So if there's no bubble in the Durham's tube, that's when you add the two reagents. The reagents test for the presence of nitrite, okay? So if you add those two reagents and your test tube turns red, that's a positive. So you know the nitrate has been converted into nitrite. Okay, so that's your other positive that can happen. If you don't get the red color when you add the two reagents, you still can't say that's a negative because it could be converted into some other nitrogen-based compound. So that's when we did that last step. We added the zinc dust. Y'all remember that last step? Okay. If you add the zinc dust and then it turns red, okay, that tells you the nitrate is still in there. The zinc dust reacts with the nitrate to give you that red color. So that confirms a negative test. So when you add the zinc dust, if it turns red, you would know it was a negative, that the nitrate was not used. Your organism does not produce the enzyme nitrate reductase. If you add the zinc dust and you don't get that red color, that tells you the nitrate's gone. So it was converted into something that you didn't test for. So this is the final positive that can occur. Okay? So you got lots of possible positives and each one tells you a different thing happened. If you get a bubble in a Durham's tube and you know it's not a fermenter, that means the nitrate was broken all the way down to nitrogen gas. If you add the two reagents and you get a red color, that's another positive. That tells you the nitrate was broken down into nitrite. Okay? It's not red, then you add the zinc dust. If it doesn't turn red, that tells you the nitrate has been broken down. It was simply broken down into something you didn't test for. But it's gone. It's no longer in that test tube. Okay, so that's a third positive that you can get. So our only negative is when it turns red when you add the zinc dust. So that is the only thing that confirms a true negative test. Okay? So any questions about that one? Okay. The last one is the citrate utilization test. So the citrate utilization test is a test that determines if the organism can use citrate as its sole source of carbon. Now this is a little bit tricky because what you're actually visualizing when you look at that test tube is not the utilization of carbon. What you're actually visualizing is the breakdown of ammonium phosphate for nitrogen. That's what makes the medium become basic. You remember back in chapter 7, we talked about the fact that organisms have to have a source of carbon, but they also have to have a source of nitrogen. Okay, so that's kind of an example of what we're talking about here. So the citrate is what it uses for carbon. The ammonium phosphate is what it uses for nitrogen. The enzyme that enables the organism to take up citrate is called citrate permease, or sometimes that's shortened to citrate. So those are both referring to the same thing. The pH indicator in this one is bromothymol blue, and that turns blue under alkaline conditions. And it stays green around neutral, and it turns yellow under acid conditions. So this is the one that turned the real pretty peacock blue. Y'all remember the one that was on a slant? Okay, so that's the citrate test. So if you see that deep blue color, that's a positive. What you're actually visualizing is the breakdown of ammonium phosphate for nitrogen. But you know if it's doing that, it's doing this too, because the only thing in there that the organism can use for carbon is the citrate. So if it's doing one, it's doing the other. So they go hand in hand. So even though we're not visualizing the utilization of the carbon, we know that's happening in that test tube. That's the only thing in there it can use for carbon. Okay? So really anything other than blue is a negative. So whether it stays green or whether it turns yellow, both of those are negatives. Okay? So any questions about that one? Okay. So what I might give you is a scenario with some test results based on these tests that we've been talking about. So I might tell you that you did the phenol broth series. You tested glucose, sucrose, and lactose. Your glucose turned yellow with a bubble. Your sucrose turned yellow with no bubble. Your lactose turned deep pink. Okay. You did the oxidase test, and you got no purple color within 20 seconds. You did the catalase test, and you got bubbles when you added the hydrogen peroxide. You did the nitrate reduction test, and you got no bubble in the Durham's tube. You got no red color when you added the reagents. You did get a red color when you added the zinc dust. And then you did the citrate test, and the citrate test turned blue. Okay, so those are your results. Everybody with me so far? Now, I would make a series of statements, and I would ask you to tell me if those statements were acceptable, meaning they're true, unacceptable, meaning they're false, or that's something that can't be determined. So, key is, in order to say it's acceptable, there's got to be a piece of information here that enables you to say, yes, I see where that happened. In order for you to say unacceptable, there's got to be something here that you can clearly look at that says, no, that did not happen. If it's simply not addressed, the information is not there, that's when you would say cannot be determined. Okay? So everybody with me so far? So let's take them one at a time. So what if I said, now these are our test results. What if I said this organism can utilize sucrose? Is that acceptable, unacceptable, or cannot be determined based on your test results?
With the nitrate reduction test?
Okay, is that the test I'm looking for to talk about sucrose?
I mean, no. Well, I was just wondering if you was doing this well.
No, these are your test results right here.
Oh, okay.
So these are your test results. So this is what we're looking at to identify these as acceptable, unacceptable, or can't be determined. So this organism can utilize sucrose. Is there anything there that tells me this organism can utilize sucrose? Of course, you would have won, all of these tests would have been inoculated with a single thing. Can't, convolute it and test double things. Only one thing at a time. Okay? That's why we work with pure cultures, because we can only test one thing at a time. So is there anything here tells me this organism can utilize sucrose? Another way to state this organism can ferment sucrose. Is there anything there that tells me this organism can ferment?
Yeah.
And what does?
The sucrose, the phenol red process.
Okay, I would look back to my phenol red broth, specifically my sucrose test tube, right? So yellow with no bubble. Well, if it's yellow, what does that tell me? It's fermenting the carbohydrate in that test tube. So if my sucrose test tube turned yellow, I know it's fermenting sucrose. So would everybody agree that that's acceptable? Yeah, because we got a piece of information here that answers that, speaks directly to that. So that would be acceptable. This organism produces the enzyme sucrase. Is there anything that we could use to answer that question?
That's not determined.
Don't think so?
Yeah.
What does sucrase break down?
Sucrose.
Sucrose.
Yeah.
What color did our sucrose test tube turn?
Yellow.
Yellow. So that means it's gotta be making
It's gotta be
sucrase,
making
right?
sucrase.
There's nothing else that breaks down sucrose except sucrase. So that would be acceptable. What if I said this organism can utilize mannose? Or I could have said ferment mannose.
That can't be determined.
And why can't we determine that?
We didn't use mannose.
We didn't do a phenol red mannose, did we? We didn't test that carbohydrate. So that's one of those where you would have to say can't be determined based on the results you were given. Remember, you're limited by the test results that you have. So you can't speak to anything outside the information that those tests give you. So that would be cannot be determined. If I said this organism produces the enzyme lactase, is there anything there that speaks to that?
Yeah, the phenol red lactase deep pink.
Phenol red lactose, and it turned
No, it doesn't.
deep pink?
If
So,
it was pink.
What does a deep pink mean?
Oh, evidence of peptide utilization.
Yeah, it's
Yeah.
not using
Not using it
the lactose, right?
Right.
Yeah.
So, it's not making lactase. If it was making lactase, it would be taking advantage of the lactose, okay? So we know that that is unacceptable based on our results.
So,
Yes, ma'am?
I have a question. On that one, you said it's not taking advantage of the lactose, and I thought I understood it over here. So if it were to turn yellow, it would show that the carbohydrates are present, so it's using it.
Right.
But, okay, all right, that's it.
Yeah.
That's my
Pink
question.
means it's using the
The
peptone.
peptone,
Yeah.
so it's not.
Yellow means it's fermenting whatever carbohydrates in that test tube.
All right,
So in order to know it's using the lactose, our lactose test tube would have to turn yellow. Okay. This organism produces the enzyme nitrate reductase. Enzyme nitrate reductase. What do you think? No. Why no? Why unacceptable?
We haven't used a reductase.
Hmm?
We haven't used one.
Yeah, we use the
I said
nitrate
acceptable.
test. Well, nitrate reduction test. So that's the one that involves nitrate reductase. So is there anything there that tells us that this organism produces the enzyme nitrate reductase? You remember, that's the enzyme it needs to
Bubble.
it does,
use the nitrate.
yeah, it would have to have bubbles to show that.
Well, it could have bubbles, or it could have turned red under a certain circumstance. So there are lots of things that would tell you it's making the enzyme, but what does our test result tell us?
No
The fact that
bubbles,
you
no
don't
red,
have a
so
bubble.
it's
No,
not.
We don't have, okay, let's take them one at a time. Everybody's getting confused. One at a time. No bubble in the Durst tube, so we know the nitrate was not converted to nitrogen gas. We know that didn't happen. Red color with the addition of reagents. So no, when we add the reagents, it didn't turn red, so that means nitrate was not reduced to nitrite, so we can rule that out. What does the red color when we add the zinc dust tell us? The nitrate is still there, right? So now can we say it's making nitrate reductase or not?
It's not.
It's not, or else the nitrate would be gone, right? So since the nitrate's still in there, we know it can't be making this enzyme. If it were making this enzyme, the nitrate would have been broken down. Does that make sense? So we know that didn't happen, so that's unacceptable. This organism can reduce nitrate to nitrite. Nitrate to nitrite. Remember, the reagents test for nitrite.
So
No red color with the addition of the reagents. Is there any nitrite in that test tube?
So it cannot.
It can't do that. It didn't do that. So that would also be unacceptable.
Professor McCullough, what is the C is no?
Cannot be determined. That's one of those where you don't have that information.
Okay.
So remember, there's gotta be something there that clearly enables you to say it's acceptable, or something there that clearly enables you to say it's unacceptable. If it's simply not addressed, you don't have that piece of information, that's when you say cannot be determined. This organism can break down ammonium phosphate for nitrogen. Is there anything that tells us that?
bubbles in the dirt. And we don't have any, so
Cannot be determined.
Okay, I hear
I said
something
no.
about bubbles in Durham's tubes and can't be determined.
there's no.
So what test am I looking for that involves ammonium phosphate?
Citrate.
Oh, this is
This
the
one, right?
Oh, we can't see.
So what does it mean if it turns blue?
That's positive.
It's a positive, but that also means that the
Ammonium
media is alkaline. That means it's using the ammonium phosphate for nitrogen. So that's what makes the medium turn alkaline. So that would be a true statement. So that would be acceptable. This is actually what makes it turn blue, is the utilization of that ammonium phosphate. So this organism produces the enzyme citrate permease. And what makes that acceptable? It says it. And we know if it's doing this, it's gotta be doing this too. And the enzyme that makes it possible for the cell to take up citrate is citrate permease, or citrase. Make sense? So, any questions? So that would be acceptable. What if I said this organism can use citrate as its sole source of carbon? That would be acceptable, yeah. We had several pieces of information here that already made that apparent. Let's see. What if I said, excuse me,